Let's see if I manage in 10 minutes. I'd like to tell you about a class of ecological interactions that I think is pervasive in shaping the structure of diversity, and I'll try to convince you of this in very little time and with a very charismatic group of organisms, the microbial pathogens that infect us, who have the great quality of evolving and having ecological interactions over relatively, over the comparable timescales and fast timescales relative to ours, so that we can observe all this unfolding. And what does this have to do with universality? Just the idea that if we have these classes of common ecological interactions, that, as I will show, give us some predictability and some ability to coarse-grained systems, we may be able to develop more universal theories. But of course, the ambition is to develop theory that would explain diversity where it is truly vast. And here we have these wonderful systems that I will not talk about, but that ecologists are very interested in, microbiomes, the rainforest, etc. And I'm talking here about the theory of assembly of ecological communities that would be at this interface of ecology and evolution. Of course, that there are pieces of that, but we don't have it. Is it even possible? We heard a lot about this, so I don't need to go about this whole thing about going from micro to microscopic patterns or then coarse graining, etc. I just wanted to point out that from the perspective of ecological interactions, we really have two classes of interactions: the birth-as processes that are present in any neutral theory, and then we have the ecological interactions that are specific and that lead to an underlying coevolutionary adaptation. And of course, we would like to have properties like what Jen showed us, but that we know are informative of the important underlying processes and that are ultimately informative of the kinds of responses we will have to perturbations. We can perhaps do prediction, especially about kinds of transitions. And increasingly in this space, ecologists are looking explicitly at the traits that underlie ecological interactions. And I like to think of a kind of trait that is very combinatorial in nature, and this, I think, would apply more general and occurs in very large spaces. So really, the microbes I'll mention here do it this way, and there are two examples. The first one is not from my work. It's still unpublished work from the one external faculty from the SFI Institute, Mark Lipsit and colleagues, and I find this extremely interesting work. This is on a pathogen that is very common among us, that causes pneumonia, meningitis, etc. They took advantage of an observation of regularity that the gene frequencies of what they call accessory genes appear to be at some equilibrium. And this is before perturbation, before vaccination. What are these accessory genes? They are the flexible part of the genome that is outside the core genome. These genes are not essential. They are not present in every strain. They come and go with a lot of lateral transfer. So this very non-essential genome was observed, and they went through this major vaccination event, and they took it as a possibility to sort of see, can we predict how the system responds? I forgot to say this is an extremely diverse pathogen, and this is the challenge to control. What they did was to build a model of the kind, I had the equations, but they are not here, of the kind that David showed us yesterday when he talked about those kinds of equations that map essentially selection to the environment. And here, they use of the order of 25 non-essential loci under some negative frequency-dependent selection to basically predict the changes in the abundance and identity of the strains in the transients and in the equilibrium after vaccination, and it works. this is very surprising. What is this negative frequency-dependent selection? It has to be interactions mediated by these genes, all kinds of interactions with other microbes, with viruses. It doesn't matter what they are. At this level, they can predict. So that is, to me, very interesting. The second example is from my own work, and I apologize to some of you who have heard about this parasite, because I'm very fond of this. This is Plasmodium falciparum. You think of the malaria parasite as a single parasite. In fact, in places of high transmission, in endemic regions of Africa, the immune system sees this parasite as an extremely diverse one. This is from the, as I said, the immune system, and going back to information and resources. What happens here is that as the host acquire immunity to specific kinds of parasites, so that as the host acquires information, the parasite loses resources. They are competing for this host, and the hosts are protected by this specific protection. So it's interesting here that resources and information are the same. Now, this parasite plays a game that is very analogous to what the immune system does, and we heard Melanie talk about this yesterday. From the perspective of a major antigen that the immune system sees, that is encoded here by a family of genes, the VAR genes, the parasite has 50 to 60 genes that encode the same variant. And if you ask how many variants are there in a small village in Africa, my colleague Karen Day, a molecular epidemiology, has determined of the order of a few thousand variants of genes. You can do the combinatorics. It is enormous if these things were just arranged at random. Now, the parasite recombines at multiple levels of organization. It recombines the repertoires. And Doug said yesterday, talking about innovation, that we need more than just recombination. We need true innovation. The parasite recombines genes to produce new genes. So it's recombining at these two levels. And you can say, well, if there is rampant recombination, there is this kind of diversity, forget finding any structure and any signature of ecological interactions having an important role here. Well, I don't have time to go over this work. We have found that there is limiting similarity in the repertoires of parasites in a structure that would be essentially how niches look in a huge space of variation. So here, negative frequency-dependent selection basically structures the system of coexisting parasites and allows diversity. Now, it's interesting because what kind of large-scale patterns we use to find regularities that told us about competition, we basically build networks of genetic similarity between parasites, and we're able to show that these kinds of networks can differentiate between neutral and non-neutral systems. Interestingly, we did this with the help of machine learning because we didn't know a priori what properties of these networks would actually tell us these things apart. Now, I will not go in detail. I just wanted to say this matters. This is from recent work and published work. I just want to mention that the same force, this is the same force, this negative frequency-dependent selection, acting at the level of the genes, essentially creates a threshold in the capacity of the system to accumulate diversity at the level of the pieces. So if you are a place in Africa, very difficult to control, you are somewhere over here in transmission intensity, and in this threshold quantity, we derived analytically for essentially the creation of diversity. It doesn't matter that you create diversity, you have to accumulate it. And interestingly, there is this nonlinear threshold affected by this frequency-dependent selection that it is operating at these multiple levels of organization. So it matters. If you want to control the system, if you push it here, it becomes as if it were a system that has biogeographically evolved at low transmission and low diversity, and now you can hit it really hard and perhaps eliminate, which you cannot do here where the parasite is play this combinatory game. I would sort of speculate that this kind of threshold applies to other very diverse systems. And I don't have time to go over details, but I love this crazy analogy. These kinds of negative frequency-dependent interactions, they call them density-dependent, it's the same, basically has a big space in the study of forests. I leave it there. I'll just have a few questions. This universal theory we do not have, but may be possible, should merge ecology and evolution, and of course, with a central role of the sort of relative temporal scales of these processes. Now, it's not going to look just like a nice replicator equation. It's going to be these messy parts playing together. In this work, there were agent-based models, very big numerically. We cannot even understand them. We asked machine learning to understand them for us. We came up with patterns. In this case, we use networks. And then, if you are lucky, perhaps you get something like what Mark had in his work, which was a nice coarse-graining model, predictive model. Now, there is this question I'm going to emphasize that we leave aside in some of our theories. We basically have a pool of variation with some sort of parameter. And that is just a given. That is very unsatisfying. What I'm telling you is that the same processes that are acting in the systems we are studying are those that over longer times and larger space have shaped the dimensionality of the trait space. That space is really important. And this is very big speculation, is that for the systems that are hyperdiverse, the trait space is vast, and it's not by chance. And that a lot of theory we have doesn't operate there. So we have neutral theory, and actually I think it's great, and it has a very big contribution to make. With no specific interactions, they predict many stationary microscopic patterns, so why should we bother with these interactions? I tried to show you that it may matter for some critical transitions. So my sense is that without them, we may fail to predict and understand important aspects of collapse and recovery. And let's see, I guess that's what I had to say. I want to thank my postdocs, who did a lot of the work, and then my collaborator, Karen Day, and her lab on the microbial, the sort of molecular epidemiology. And, oh yes, because we are here, I'll thank also the Ecology and Evolution of Infectious Disease program, which is together between NSF and NIH. Thank you.
The maintenance of diversity depended critically on negative frequency dependence, now, which is an obvious feature of parasite immune interactions. But in forests, presumably, it's not dominated by negative frequency dependence. Is it just frequency dependence, or okay?
Well, very good question, of course. It was asked by David. But no, I actually should say it's very interesting because for example, in the malaria system, I am talking about immunity, and therefore it's negative frequency dependence. In Mark's system, we don't know what it is. It's because it's not those genes are not encoding for the traditional serotypes. So surprisingly, it's not the main determinants of immune memory to these strains. The fate of these strains is determined by genes that are not involved in the main immune recognition. So it must be all sorts of, I cannot tell you what, interactions that are clearly mediated by these genes we think are not important. And now getting to the rainforest, people have looked at this. to some degree, and of course there is the all very influential Johnson-Connell hypothesis of interactions with specific enemies, so it should be negative in that case. But of course we are learning a lot about how those are a combination of mutualisms and also interactions with enemies. There was a fascinating paper this year that looked at the traits that underlie these kinds of density-dependent effects, and they found that it was not the traits that have to do with growth and resources, it was the traits that have to do with a lot of chemical, aspects of the chemical phenotype of the interactions with herbivores, which may be a lot more labile and a lot more able to change evolutionary in short time scales.